The control of blood glucose levels is incredibly important. And although the pancreas gets a lot of attention due to its hormonal roles in secreting insulin if the glucose levels get too high, and secreting glucagon if the glucose levels get too low, those signals are actually controlling other tissues, especially your liver, and allowing the liver to take in extra glucose if the blood glucose is too high, or to release glucose if the blood glucose is too low. And a lot of the ability of the liver to do this is thanks to special properties of its glucose transporters, as well as of its hexokinase enzyme, which is referred to as glucokinase, the version that's in the liver. These versions of these transporter and of this hexokinase enzyme that the liver has are especially suited for the liver's roles in helping to regulate the blood glucose levels. Unlike most of your tissues, the liver has glucose 6 phosphatase, which is going to allow the liver to actually release glucose to the body. And it also is able to store glucose that it takes in as glycogen, as well as transform it into fatty acids, which it can then store as triacylglycerols or tags. And so the liver has really, really important functions, and the enzymes and transporters that it has help it carry out these functions. So let's see how the liver's hexokinase, and so the version of hexokinase in our liver is called glucokinase, as well as how its glucose transporter helps it to be able to carry out these important tasks. Because not only does the liver get signals about blood glucose levels from hormones like insulin and glucagon, but it also is able to directly sense blood glucose levels thanks to the actions of its transporter and of its hexokinase. So let's dive in. First off, let's orient ourselves. So when we think about glucose coming into the cell, there are several different fates for that glucose. It can go in glycolysis, it can get stored as glycogen, it can go the pentose phosphate pathway, lots of different options. But the first thing is just to keep the glucose inside of the cell. So the glucose is going to come in through these transport channels we'll talk about in a second. But those are channels, it's not active transport. And so what's happening is that if the glucose comes in, but it doesn't get trapped, it can go back out if the concentrations inside the cell get higher than the concentrations outside the cell. But those channels are for glucose and not for glucose 6 phosphate. And so if you phosphorylate the glucose, now you're trapping the glucose inside of the cell. And then you're also lowering the glucose concentrations inside the cell because you're now changing the glucose to glucose 6 phosphate. This is an irreversible step, and so in order to go around it, you have to use a different enzyme to remove that phosphate group. This different enzyme that's used is glucose 6 phosphatase, this G6Pase, but only select tissues, notably your liver and your kidneys, actually have that glucose 6 phosphatase, this G6Pase enzyme. So most of your tissues, if they phosphorylate the glucose, they've kind of committed themselves to keeping it. Therefore, it's important that we have mechanisms that allow the tissues to get the glucose that they need and trap the glucose that they need, but not trap too much. So as we'll see, we're going to get, for most tissues, you'll get feedback inhibition, where this glucose 6 phosphate, this G6P, is actually going to inhibit the hexokinase that's phosphorylating it. But the version that we have in our liver is not going to be inhibited by the glucose 6 phosphate. And this allows our liver to sort of serve as a failsafe and take in more glucose because the liver can store it for later. So let's go in a little more detail, starting with glucose transporters. Our bodies have multiple glucose transporters, these GLUT transporters. And we're going to focus on kind of, for now, just GLUT2 and GLUT3. GLUT2, this is going to be found in your liver, your pancreas, your intestine, and your kidneys. And GLUT3, this is going to be found in your brain. If we think about what the liver, pancreas, kidneys, and intestine have in common, well, they're all involved in blood glucose regulation. Your liver and your kidneys, as we talked about, can release glucose to the bloodstream. Your liver can release glucose from glycogenolysis, so breaking down glycogen, or from gluconeogenesis. Your kidneys can release glucose from gluconeogenesis, but they don't store glycogen, so they can't release it from glycogenolysis. But both tissues are able to release the glucose because they have the glucose 6 phosphatase, the G6Pase. Your pancreas and your intestines are important for secreting hormones that help those liver and the kidneys and your other tissues know what to do in response to blood glucose levels and know the glucose levels in a sense indirectly. But you also want these tissues to be able to directly sense the glucose levels. And so they need to know what the glucose levels are out in the bloodstream. And the way that they can do this is by having a low affinity glucose transporter. So let's think about this. The GLUT2 is going to be lower affinity than the GLUT3. And how do I know this? I look at the KT value. The KT, this is like a Km value for an enzyme, but the T is for the transport. So the KT value is for the glucose concentration, or whatever the substrate is, in this case it's glucose, concentration at which the transporter is transporting at half its maximum velocity. So a lower number is going to equal a higher substrate affinity. And so the lower number of a KT value is going to indicate a higher affinity for glucose. If we look at GLUT2, we see it has a KT of 17 millimolar. Whereas if we look at GLUT3, we see it has a KT of 1.4 millimolar. Therefore, the GLUT2 has a higher KT value. It has a lower affinity than the GLUT3. If we think about kind of physiological levels of glucose, so they're going to be somewhere in this range typically. For like fasting glucose, it's about 5 millimolar. So you're somewhere over here. At those conditions, the GLUT3 transporter is going to be working at its maximum. It's already saturated. But GLUT2, you're still in this linear range. So if you're here and then you increase the glucose concentrations, your transport rate isn't going to change if you had the GLUT3 transporter. But if you have the GLUT2 transporter, now your transport rate is going to change. And thus your cell is going to be able to sense that there are changes in the blood glucose concentrations. Here, it's basically like you have an overflowing cup. You can't tell if you're adding more water because the cup is already full. You fill up your cup really quickly, and then you can't tell if anything else is happening. Whereas with GLUT2, here you're actually able to see those levels in your cup changing. And so GLUT2 is in this linear zone. It's able to sense those levels, and thus the inside of the cell is going to reflect the extracellular concentrations of that glucose better. So it's important that these tissues are able to sense it. But what about the tissues that it's like, I don't care if I can sense it. I can't even do anything to control the blood glucose levels. I don't have G6Pase. I don't secrete these hormones. I just want glucose. So one of those tissues like that is your brain. And so your brain, it just really needs that glucose, especially because the blood-brain barrier is going to prevent the brain from getting fatty acids for energy. So it's going to really rely on that glucose. So you want it to be able to take in glucose, all the glucose that it needs, even if the glucose levels in the bloodstream are really low. And so you want it to have this GLUT3, which has this really high affinity so that it doesn't take that much glucose and it's already transporting at its maximal rate. And then it will allow the brain to get the glucose that it needs. And so having this GLUT3 transporter is going to help the brain get the glucose that it needs. But if the brain has such a high affinity glucose transporter, what's going to prevent it from hogging all the body's glucose? Feedback inhibition. So now we need to think about the next step of our pathway. Because getting the glucose into the cells is great, but if you don't trap that glucose, well, now the glucose is just going to leave the cells if the glucose concentrations inside the cell get higher than the glucose concentrations in the bloodstream. And so we need to make it so that the brain doesn't take too much. And the way that we're going to do this is by making sure that we don't trap too much. The trapping, remember, is done by this hexokinase enzyme that phosphorylates glucose at the 6th position to give you glucose 6-phosphate or G6P. What's going to happen is that if the tissues are taking in glucose, they start phosphorylating it and trapping it. If they trap more than they need, the G6P is going to build up. When the G6P builds up, it's going to inhibit the hexokinase enzyme. So it's going to bind to the hexokinase enzyme and actually prevent the hexokinase from phosphorylating more glucose. This is going to prevent the cells from trapping in any extra glucose that they take, and so that glucose would then just leave the cell. So this allows the brain to take in all the glucose that it needs, ensure it has enough that it needs, but not take too much. So this is kind of a similar looking chart to what we saw before, where we want to think about when we're in the linear range and when we're saturated. But here we're looking at hexokinase rather than looking at glucose transporters. This black line is our hexokinase 1, which is the version of hexokinase that we have in most of our tissues. We'll talk in a minute about this hexokinase 4, also known as glucokinase, which is the version that we have in our liver and things. What's going to happen is that you have a high affinity hexokinase in these tissues with the hexokinase 1. So they're going to be able to quickly trap the glucose that they take in, readily trap that glucose, just like they were readily able to take in the glucose. However, they're going to then get inhibited by the G6P if they start taking in too much. And therefore, they're going to plateau at a lower velocity. So note that in that graph before, that was just like a sketchy graph that I had that was doing it based on normalized initial velocity. So it's maximum, but it wasn't specifying what that maximum was. Here we can see that the maximum rate of the phosphorylation of hexokinase 1 is going to be lower than that of hexokinase 4 because of the hexokinase 1, we're actually going to be inhibiting it with our G6P. And so we start taking in the glucose through that glucose transporter. We trap the glucose, but now we've trapped too much. What are we going to do? We don't want to take all the glucose from the bloodstream. Don't worry, the glucose 6-phosphate is going to build up, and that's going to inhibit your hexokinase 1. That's going to keep the brain from taking in too much, but it'll allow the brain and other tissues to take in what it needs. It won't, however, allow the brain to sense the glucose levels, but we don't really care because the brain can't do anything about it. But some tissues can do things about it. So your liver, say, it has this hexokinase 4. This version is also called glucokinase, and it's also found in your pancreas. It has kinetic properties and regulatory properties that are related to its role in maintaining glucose homeostasis. So remember that your liver is actually able to release glucose, and you want it, therefore, to be able to release glucose without phosphorylating the glucose inside of the cells that it's trying to let go of. So you want to make sure that you don't have too high of an affinity of the hexokinase because you don't want to be trapping the glucose that you're trying to let out. You also want to make sure that you don't have too high affinity because you don't want the liver to be taking in too much glucose if the other tissues don't have enough. So those other tissues like your brain, they need to take first dibs, but then you want your liver to be able to pick up the slack if there's leftovers. So what's going to happen is that this hexokinase 4, as we discussed, it's able to reach a higher level of activity. So it's able to reach a higher rate of phosphorylating the glucose. And it can store that glucose, remember, as glycogen. It can store it as fatty acids and lipids. So it's okay if it takes in a lot, as long as the other tissues have what they need. It needs to be able to sense the glucose levels, just like we saw with it being able to sense the glucose levels through the transporters. And so again, we see that in these physiological levels, we're in the linear activity range of this enzyme. So as the glucose levels change, the rate of phosphorylation is going to change, and thus the liver is kind of able to sense the glucose levels. And because it's a lower affinity than the hexokinase 1, we're going to prevent the liver from taking all the glucose. The other tissues take what they need, and now the liver can take the leftovers. It's important that you don't let the glucose build up in the bloodstream. And so this hexokinase 4 is not going to be inhibited by glucose 6-phosphate. This is going to make it so that the liver can take in more than it can kind of handle at the moment. but the liver is able to store it. And so it's able to take up the slack when your other tissues have taken what they need, but there's still high blood sugar left over. So what happens is that when you eat, the glucose kind of first goes through your liver, and your liver takes some initially and then sends the rest to other tissues, and the other tissues take what they need, and then the excess goes back to the liver, and the liver can then store the extra glucose. And it's able to store it because it has the enzymes to hook it up and things, but also because it's not getting inhibited by the G6P buildup. So there you go. The liver is really important for helping control blood glucose levels. If blood glucose is too low, it's going to make glucose via gluconeogenesis and or free up glucose from glycogen via glycogenolysis and release it to the bloodstream. Unlike most tissues except the kidneys, the liver has glucose 6-phosphatase, G6Pase, so it's able to dephosphorylate and thus untrap and release glucose to the body. So remember that even though your muscles and your liver both store glycogen, when your muscle breaks down glycogen, they can only take it to glucose 6-phosphate. They can't remove that 6-phosphate group. And so they can use the quote unquote glucose, really the glucose 6-phosphate that they release for themselves, but they can't actually give it to other tissues. Whereas your liver is able to give it to other tissues. Your liver is also able to make that glucose from scratch through gluconeogenesis, as is your kidneys, and they can then release that glucose to the bloodstream. But the glucose in your bloodstream, if it's not coming from food you ate, if it's coming from your liver, it could be from glycogenolysis or from gluconeogenesis, whereas that coming from your kidneys can only be coming from gluconeogenesis because your kidneys aren't storing the glycogen. If your blood glucose is too high, the liver is going to take in that glucose and store it as glycogen via glycogenesis or fat. So how does the liver know what to do? It needs to be able to sense the glucose levels. It can do that through hormonal cues from insulin if the blood sugar is too high, and glucagon if the blood sugar is too low. Those are released from your pancreas, so your pancreas also needs to be able to sense blood glucose levels. These hormones are going to trigger signaling pathways that regulate the enzymes involved in these various processes. The liver is also able to directly sense glucose levels thanks to low-affinity glucose transporter and hexokinase 4, which is also known as glucokinase. In these ways, the liver is able to help regulate the blood sugar levels in combination with other tissues. And there are multiple levels to how this happens. And a couple of these are going to be through the glucose transporters and the hexokinase.